Hello, I am your teacher for today, Ma'am Amy Rose T. Calma. We are going to discuss the trace elements. Here are the following objectives of the study. At the end of the module, you will be able to describe the different metabolic functions as well as the significant trace elements in the human body. The following are the important parts in the discussion of the module. The first one will be the introduction. This will give you an overview of the Module 6. The second one will be the discussion about the biologic function of the trace element. These are important functions of the trace elements that are found in the body. It will then be followed by the third part, which is all about the specimen collection and processing. This will provide knowledge on how a specimen is to be used, collected, and stored, most specifically for trace elements. The fourth part will be the methods and instrumentation. You will be able to determine the important laboratory instruments in order to evaluate the presence of an essential or a toxic element. The following instruments will be discussed, namely atomic emission spectroscopy, atomic absorption spectroscopy, and inductively coupled mass spectroscopy. The fifth part will be the interferences. These will equip you of the commonly encountered interferences in the laboratory and what to do when this problem arises. The sixth part will be the elemental speciation. It involves the determination of the chemical forms of an element in a particular matrix. The last part will be the discussion of the essential and non-essential trace elements. These are commonly found in the body. Let's begin with the introduction. Essential and non-essential trace elements should be defined, and they are as follows. When we talk about an essential trace element, it is considered essential if a deficiency impairs a biological or a functional process, and the replacement of the element corrects this impairment. In Tagalog, kapag sinabi po natin ang essential, kapag nawala sila sa katawan natin, magkakaroon tayo ng deficiency or magkakaroon tayo ng sakit. Pwedeng masabi natin na essential itong bagay na to kapag pinalitan or by means of a substitution, mawawala itong ating pong sakit. So, we call this element an essential trace element. When we talk about essential trace element, makikita rin natin ito sa pagkain. These are important because they are also found in our diet. Associated also with an enzyme and other protein as a essential component or cofactor. Dito sa part na to, sinasabi na ang ating pong mga enzymes ay importante, essential sila kapag tumutulong sila sa mga enzymes natin para mapabilis ang isang chemical reaction. And so we call these enzymes or we call these metals or trace elements as a cofactor. These elements are found in small quantities such as milligrams per liter para kay iron, copper, and zinc. Alam naman po natin na kapag sinabi natin ng iron, this is involved, in the regulation of our red blood cells, most specifically with the oxygen content that is found in the hemoglobin. Same as true with copper. Tutulong din ng copper natin sa pagpaprocess ng ating pong iron component na makikita po natin sa red blood cells that is found also in the hemoglobin synthesis. Zinc. Lagi natin nakikita itong zinc na to sa ating pong mga vitamins because one of the important function of our zinc is it boosts our immune system. So these are examples of essential trace elements. Ultra trace elements, on the other hand, are extremely small amount in our body that is less than micrograms per liter. We also have the non-essential trace elements. Non-essential kasi they are of medical importance. And many of the non-essential trace elements, once they enter our body, will become toxic. Okay? Or pwede po itong mag-cause ng sakit sa atin. Conditions that can result in deficiency of several trace elements might be because of decreased intake, impaired absorption, increased excretion, and genetic abnormalities. So kung titingnan natin ito sa Tagalog, makikita natin na minsan nagkakaroon tayo ng trace element deficiencies, okay, dahil nagkulangan ka sa diet, okay? So for example, there are some trace elements that are found in meats, pero paano kung for example, ikaw ay isang vegetarian, di ba? So pwede kang magkaroon ng kakulangan sa trace elements na yon dahil decrease ang intake mo when it comes to your diet. It might be also because of a problem in the absorption. Okay, for example, in the small intestine, we have the duodenum, jejunum, di ba? Ileum. Okay, paano kung halimbawa meron kang problem dito sa iyong gastrointestinal tract and therefore, it would lead to a deficiency in a particular trace element. Thus, there will be an impaired absorption. Increased excretion. So, paano naman kung halimbawa sobrang napabilis, 'di ba? Ang pag-excrete natin dito po sa ating pong mga essential na trace elements, 'di ba? Baka sobrang bilis nito mawawala na sa loob ng katawan natin, ne? This is brought about by increased excretion through the feces or through the urine. This will also lead to a deficiency. And lastly, some of the problems that are involved in the deficiencies is genetically acquired. So they may be autosomal recessive or autosomal dominant na pwede rin pong mag-lead sa ating pong mga deficiencies when it comes to the essential trace elements. The essential trace elements are often associated with an enzyme or another protein. So kagaya po na nabanggit ko kanina, kapag ang ating pong essential element, most of our essential elements are metals. Kapag ang metals is naka-attach siya or bounded po ito sa ating pong mga enzymes, ang tawag natin sa kanila metalloenzymes. When this happens, they give an important function, again, with regards to our chemical reaction. Yeah? And also, essential trace element may be a protein. At kapag naman po yung ating pong important metals, ang mga metals na to kasama po in the presence of the metal and the protein, then your metal will act as a cofactor or otherwise known as a metalloprotein. Deficiencies typically impair one or more biological functions. Excess concentration naman, on the other hand, pag napasobrahan po, ito po ay maglilid sa atin ng toxicity. Some examples of the trace elements involve iron, copper, and zinc. Ultra-trace elements involve selenium, chromium, and manganese, which are found in microgram or microgram per liters or parts per billion concentration. So, sobrang liit po ng ating po mga ultra-trace elements. Let's talk about the biological functions of the trace elements. Catalysts. Again, I have mentioned this a while ago. When we talk about catalysts, they speed up chemical reaction. Now, one of the characteristics of our trace elements is that they are what we call as metalloenzymes. Parang kanina po nabanggit natin na kapag yung ating pong mga enzymes ay laging nakabound sa ating pong mga metals, then ang tawag po natin sa kanila metalloenzymes. At nakakatulong po sila para mapabilis ang isang chemical reaction. Next, they participate in the oxidation reduction for energy metabolism. So some metallic ions such as iron and copper participate in the oxidation reduction reaction in the energy metabolism. We have discussed this in our previous chemistry subjects kung saan ang oxidation, meron tayong Leo. Oxidation, there will be loss of the electrons and gain of electrons for the reduction. One of the characteristics of our metals is that they are proton Di ba, yung mga metals po natin, di ba, these are positively charged. They are cation. So therefore, sila po ay nagbibigay, di ba, na binibigay nila yung mga electrons nila. They are donators of protons or electron donator, di ba? Mapagbigay ang ating po mga metals. Non-metals, on the other hand, sila naman po, di po ba, sila ay nagsishare ng mga electrons para pareho silang kumpleto. And so, one of our good characteristic or biological functions of our elements, of our trace elements, is that they participate in the redox reaction, which is important for energy metabolism. And this is with regards with the production of the ATP, which is also the third role in the biological functions. This is most importantly on the part of the iron. Iron is a constituent of hemoglobin and myoglobin and plays a vital role in the transport of oxygen. Ayan. So as we all know, our iron is very important because it is again part of the hemoglobin synthesis kung saan hinahawakan po niya yung ating pong oxygen. Diba? So it is found in the hemoglobin. And so, pwede din po natin makita itong iron sa ating pong myoglobin kung saan makikita naman natin ito sa mga muscles, which plays a vital role in the distribution of oxygen in the different parts of the body. So, as what I have mentioned a while ago, our biologic functions include also the elements, particularly the metals, wherein they are bounded by our enzymes, which we call metalloenzymes. In here, it was stated that enzyme activators, these are chemical compounds that increase a velocity of enzymatic reaction. Some metals help the enzymes speed up a chemical reaction. Kaya nga po nabanggit natin na kapag ang mga metals na to, kapag sila po ay nakabound sa ating pong mga enzyme, ang tawag natin sa kanila is metalloenzyme. So kapag sinabi po natin ng mga metalloenzyme, they are the ones that are bounded to our enzymes. Okay, yung mga metals natin, bounded po sila sa mga enzymes natin. They are also known as a cofactor. On the other hand, when the metal helps increase the activity of the enzyme, they are called metal-activated enzyme. As you can see in the picture, here is a distinction between the metalloenzymes from that of the metal-activated enzymes. In here, we have the definition. As we all know, for the metalloenzymes, they are enzymes that are tightly bound to the metal ion. Whereas for the metal-activated enzymes, the metals are enzymes that have increased activity because of the presence of the metal ions. Attachment to a cofactor. A metalloenzyme is firmly bounded to a metal ion, and this is the cofactor. Whereas for the metal-activated enzyme, they have no firm bound to the metal ion, or it is not firmly bounded to a metal ion. One of the requirements for the activity for the metalloenzyme is that it requires either one or two metal ions that is bound to the specific region of the enzyme. For the metal-activated enzyme naman, it requires high concentration of metal ions around them. So, hindi po niya kailangan na laging naka-bound, okay, or naka-attach sa ating pong enzyme, yung metal. So, dito, kahit maraming contents lang, okay, it should require a high concentration of the metal, okay, around them. Pero pagdating sa metalloenzymes, ito po, kailangan nakabound talaga sila. Okay, doon sa enzyme yung mga metals. Example of the metalloenzymes, we have thermolysin, diol dehydrolase, cytochrome C oxidase, peroxidase, and arginase. Some of the examples of metal-activated enzymes, we have pyruvate kinase and phosphotransperases. The biological functions includes also the electron transport chain. The electron transport chain is a series of 4 protein complexes that couple redox reactions creating an electrochemical gradient that leads to the creation of ATP in a complete system named oxidative phosphorylation. It occurs in mitochondria in both cellular respiration and photosynthesis. Dito sa slide na to, makikita natin ang isang napaka-importanting function ng ating pong mga trace elements kung saan sila po ay involved sa pag-process ng energy ng mga cells natin. Ito ang tinatawag nilang electron transport chain. As we also see in our slide, makikita natin na the powerhouse of the cell is the mitochondria. And so therefore, dito po yung paggawa po natin ng adenosine triphosphate. Tumutulong po ang ating pong mga essential trace elements sa paggawa at pagpaprocess para magkaroon po ng enough energy ang bawat isa sa mga cells po natin. And so this is what happens in the electron transport chain. Similarly, makikita rin po natin dito that iron is an important metal that participates in the electron transport chain. Next, we proceed with the specimen collection and processing. Specimen for analysis of trace elements must be collected with scrupulous attention to details such as the anticoagulant, collection apparatus, and the specimen type. This usually includes the urine, serum, and plasma or the blood. Because of the low concentration in biologic specimens, extraordinary measures are required to prevent contamination of the specimen. This includes the use of specific sampling and collection devices with the specially cleaned glasswares and water and reagents that are of high purity. Dito, mga anak, makikita natin na kailangan makontrol natin yung mga environmental factors na pwedeng mag-cause ng inaccurate results natin. As we all know, ang ating pong mga specimen ay trace element. Ibig sabihin sila po ay napakakonti. They are very minute na kapag meron pong mga uncontrolled variables, so hindi po natin maiwasan na makikita sa environment such as the air, or the dust particles, o yung pong mga iba na pwedeng constituents ng ating pong mga test tubes, pwede palang mag-cause ng false or inaccurate results sa ating pong mga specimens. Kaya kailangan bigyan po natin ng scrupulous attention itong mga to. The selection of needles, evacuated blood collection tubes, anticoagulant and other additives, water and other reagents, pipette, sample cups must be carefully evaluated for use in the trace and ultra-trace analysis. It is also recommended that in the laboratory environment, placing the trace elements laboratory in a separate room, incorporating rigorous contamination control features. So napaka-importante mga anak na as much as possible, kung pwede sana ilagay natin sila sa separate room para mas controlled natin ang environment natin. Such controlled features contains placing a sticky mats at the doors. Ayan. Some of the essential trace elements may be acquired through gas, through air. So therefore, itong mga to kailangan as much as possible makontrol natin. And one way is that we place sticky mats in the doors. Non-shedding ceiling tiles, ayan, baka po mahulog po yung ibang mga elements na nandyan sa paligid natin, kagaya ng mga nasa kisame o yung mga ceiling tiles natin na pwedeng mag-cause ng inaccurate results. Carefully controlled airflow to minimize the particulate contamination. Again, some of the essential elements can be acquired airborne, and so therefore we also have to control the airflow. Disposable booties, worn over shoes, and particle monitoring equipment should also be done. For the specimen collection and processing, we have to collect in a manner to minimize the contamination of the other trace elements that are not found in the specimen. And so therefore, kailangan po natin dito yung royal blue EDTA tube. Ayan, okay? So this is the specimen or the test tube that is ideal in collecting the trace elements. Royal blue trace element serum tube. The use of the stainless needles. And pag kumukuha po tayo ng mga specimens, unang-una po natin kuhanin yung ating pong trace element. The trace element specimen should be the one to be first that is to be collected prior to the other tests. For the methods and instrumentation, the commonly used instrumentation for trace elements and toxic metal analysis is the atomic absorption spectroscopy. Atomic absorption spectroscopy is also known as your atomic absorption spectrometer or atomic absorption spectrophotometer. So, pareho-pareho lang po sila. So, with regards to this, they can either be making use of a flame or without a flame. So, kapag gumagamit po tayo ng flame, this involves the flame atomic absorption spectroscopy. Kapag naman walang flame, this makes use of the graphite furnace atomic absorption spectroscopy atomization. So, this one is flameless. There are also other methods such as we have the atomic emission spectroscopy. So, sa atomic emission spectroscopy, madalas po ginagamit po ito kasabay ng ating pong tinatawag na inductively coupled plasma atomic emission spectroscopy. So, pag minsan, makikita po natin ito sa mga test po natin o sa mga instruments that are used to determine the presence of the trace element. Madalas po, ang ginagamit po, pinagsasabay si atomic emission spectroscopy at saka si inductively coupled plasma emission spectroscopy. Ito pong mga to is for atomization and excitation. So, yan po yung mga ginagawa po nila. Madalas, pagka si atomic emission spectroscopy, pinipair siya kay ICP o yung tinatawag nilang inductively coupled plasma atomic emission spectroscopy. Meron din si actively or inductively coupled plasma mass spectroscopy, sinasama din siya dito kasi mas sensitive siya, mas marami yung mga elements na kaya niyang itest at medyo free siya from the interferences o yung mga problems na na-encounter natin when it comes to the collection of the specimen, most especially sa mga trace elements. Again, the inductively coupled plasma mass spectroscopy is more preferred than the other test kasi this one is very sensitive. It has a wide range of elements that are covered and it is relatively free from interferences. The first one, we have the atomic emission spectroscopy. So, the atomic emission spectroscopy is otherwise known as your emission or atomic emission spectrophotometry. It is also called emission flame photometry. So, kung ano po ang nangyayari, unang-una, yung ating pong metallic salt, okay, kapag ito ay na-ignite making use of a flame, it will be excited. Ayan, so this will make the electrons more excited, thus they will go out of their orbits. As the excited electrons return to the lower ground, they emit electromagnetic radiation. And then, so pagka-emit po nila ng electromagnetic radiation, ito po ngayon ay mababasa. Okay, the heat energy absorbed and the light emitted are now the characteristic atoms under consideration. So kung ano po yung gusto po nating mabasa o gusto pong i-absorb nung particular na trace element na yon, yun po yung makikita natin under the detector. And so, you have to remember that each metal has its own emission spectrum showing emission at characteristic wavelengths. So, taking a look at our picture, makikita natin na yung ating metallic salt. Ayan, di ba? Kapag daw ito ay flame, nag-undergo ng flame, it will excite the electrons. As they excite the electrons, they are going to emit electrical charges. So, and then, bababa siya sa ground state. Noong nag-excite ng electrons po natin, naglabas ito ng electromagnetic radiation at ito po ay nabasa sa ating pong monochromator. Depende sa ating pong trace element na babasahin niya, pipiliin ng monochromator kung sino lang po yung gusto nating tignan na trace element. From there, magse-select po ang monochromator at isa lang po ang lalabas sa detector. The rest Okay, the other elements that are not red o na hindi po natin kakailanganin sa ating pong trace element will not be detected. And so therefore, ang lalabas lang po dito ay isang element lang. So kung ano lang po yung gusto po nating ma-detect, then that is the corresponding characteristic wavelength that will be read by our detector. So there are three most important components in the atomic emission spectroscopy. The first one, we have the source. Ayan. Source, this is in which the sample is atomized at a sufficient temperature to produce an excited state species. Those species will emit radiation upon relaxation back to the ground state. So kagaya na nabanggit ko kanina, kapag ito po yung ating pong metallic salt ay na-ignite, the electrons will be excited, and after po nila na-excite, bababa po sila at the ground state. Now, what is important here is that the gas is the source of the flame energy. This includes a mixture of hydrogen and oxygen gas, or it also be a natural gas, acetylene, a propane, in conjunction with the air and the oxygen. The flame temperature should be held constant because it affects the sensitivity and the response to flame photometers. So ito yung pinaka-importante natin. Ang unang-una is the source. Then we have the wavelength. The wavelength selecting device or the monochromator for the spectral diversion of the radiation and separation of the analytic line from the other radiation. This will isolate specific wavelength of interest. Kagaya na nabanggit ko kanina, upon the excitement or excitation of our electrons, and then they will be in the ground state, mag-e-emit po ito ng electromagnetic radiation at mababasa ito ng monochromator. Ito ngayon ang pipili kung anong wavelength of interest ang kukunin natin. So, that is the second important component of the atomic emission spectroscopy. Then we have the detector. This permits the measurement of the radiation intensity. The detectors require water and standards to establish thermal equilibrium before measurements are taken. Ayan. So makikita natin sa detector, isa lang po yung babasahin niya nang galing sa ating pong monochromator. These are the three important components in the atomic emission spectroscopy. We have the source, the monochromator or the wavelength selecting device, and the detector. Typical fuel gases that are being used in atomic emission spectroscopy, we have the flame and inductively coupled plasma. Ang mga ginagamit po natin ay ito, typical fuel gases such as the hydrogen, acetylene, and oxidant gases. Namely, we have the air, oxygen, and the nitrous oxide. So, meron din po tayong mga gas chambers na present dito sa ating pong atomic absorption. So, for gases, they are combined in a specially designed mixing chamber. So, first one, meron tayo ditong nebulizer. The nebulizer is the one that converts the liquid into a fine spray. So, ito po yung nebulizer natin. Siya po yung nagko-convert ng liquid into a fine spray. Then, you also have to remember that in the atomic emission spectroscopy, both the atomic and ionic excited states can be produced depending on the element and the source. This will lead to the production of a complicated emission spectra. Thus, kapag nakagawa na po siya ng spectra, ito po yung ating pong line o yung mga fine lines po natin. Emission spectra or the emission spectrum of element is composed of a very narrow peaks. These are sometimes called lines. With each of the line with a different wavelength and each line match with a specific transition. Ayun po yung mga sample po ng ating pong mga emission spectra that are found in our atomic emission spectroscopy. Each element has their own characteristic emission spectrum. So for example, our sodium, makikita po natin siya with a wavelength of 589 nanometer. So pag minsan nag-overlap po sila resulting to interferences. Kaya ang madalas na ginagawa po natin para maiwasan natin ang mga wavelength na to is to make use of a limit as a quantitation, freedom from interference, and robustness. So para ma-overcome natin ito, we may make use of a chemical modifier. Other interference naman, yung ating pong flame hindi niya kayang mag-dissociate with the sample. Di ba? Kailangan napaka-importante na dapat ma-dissociate o ma-pyrolyze, di ba? When it comes to the presence of the heating process. Pero nagkakaroon ng chemical interference kapag hindi nangyayari yon. Then we have the ionization interference kung saan yung ating pong mga samples nagiging excited lahat sila. And lastly, we have the matrix interference kung saan yung light na absorb sa organic substance and then yun nga yung light absorption niya. So, by the presence of the organic substance, ma-enhance yung absorption niya doon sa light. Minsan naman, yung nangyayari dito, yung upon exposure of the sample or the specimen sa flame, ang nangyayari, nagpo-produce ito ng solid particle. And so, this pertains to the matrix interference. Then we have the inductively coupled plasma mass spectrometry, or we also call it the inductively coupled plasma mass spectroscopy. So, kahit alin sa kanila, pwede. So, for the inductively coupled plasma mass spectrometry, so this is a state-of-the-art analytical technique for elemental analysis. The term plasma na ginagamit po natin sa inductively coupled plasma, ito yung paggamit natin ng ionized gas, si argon, in which a certain proportion of electrons are free. Like other mass spectrometer, the ICP-MS or the inductively coupled plasma mass spectrometry makes use of a mass to charge ratio. So, yung ating pong M dito sa ating pong formula na to o yung ating pong ginagamit natin dito sa molecular mass divided by the ionic charge wherein the M is your charge and the Z will be the ions. Ayan. So, yun po yung ginagamit po nating unit para sa ICP-MS or inductively coupled plasma mass spectrometry. So, tinitingnan natin yung ratio ng ating pong charge doon sa ion or the mass to charge ratio o yung tinatawag nilang M over Z. M meaning charge and Z meaning ions. This is the picture of a ICP-MS or inductively coupled plasma mass spectroscopy or spectrometry, wherein the following components are essential. The first one, ion source. Ion source is the part of the mass spectrometer that ionizes the material under analysis. So kung titingnan natin ang ion source natin ito. Yan. So this is the ion source. This will ionize the material to be analyzed. The ions are then transported by the magnetic or the electric field, which is the mass analyzer. So, ito yung mass analyzer natin. So, the mass analyzer is the component of the mass spectrometer that takes the ionized masses and separates them. Yan, pinaghihiwalay po sila according to their charge to mass ratio and outputs them to the detector, which is our third important component. So the ion detector, this measures the value of the indicator quantity and thus provides a data for calculating how much ion is present. Ayan, so yung ion detector po natin, ayan dito po. Ayan niya. So dinedetermine niya kung gaano kadaming mga ions ang present. So the important components, again, we have the ion source niya, kung saan po siya yung nag-ionize ng material for the sample to be analyzed, followed by the mass analyzer, pinagsiseparate po niya according to their charges. And then afterwards, we have the ion detector. Depende po doon sa gusto po na i-measure na ion. Yun po yung ilalagay po niya doon. Then we also have the process. So the argon plasma induced by commercial o yung ICP natin generates a temperature, napakataas po ng temperature. This is from 6,000 to 10,000 Kelvin. And it will serve, marami po siyang purpose o marami siyang gagawin. Yeah? Then, unang-una, kaya mataas po yung temperature niya, ida-dry niya. And then afterwards, hindi lang po i-dry, i-vaporize pa, di ba? So, and then the droplets will be vaporized by the nebulizer. So, kumbaga, hindi lang po ito papatuyuin. Gagawin pa siyang vapor, di ba? So, it will turn it into gas. Therefore, napaka-importante po na mataas ang ating pong temperature. This step is followed by atomization of any molecular species. And finally, the atoms are thermally ionized, at which point they are ready for the introduction into the mass spectrometer. Ayan. So, napaki-importante po na yung ating pong process dito, simula, the argon plasma, will generate a very high temperature para po ma-dry at ma-vaporize yung ating pong mga droplets that is being produced by the nebulizer. And then afterwards, atomization occurs. And finally, they will become ions, kung saan po imi-measure ng ating pong instrument yung number of ions that are present based on the given element. So, this is an example again of a simplified schematic diagram of the ICP-MS or inductively coupled plasma mass spectrometry. Nearly all of the ICP torches consist of 3 concentric quartz tubes that surrounds a coil carrying radio frequency power. Ayan. So, nasan ba yung torch natin dito? Ito yung torch. Ayan. Ito po yung torch natin. ICP torch. The middle tube of the torch will carry the argon. Diba? Ito yung plasma. Yung sinasabi ng ICP o yung plasma na pinapertain ng ating pong instrument is the argon. Diba? This will form the plasma. Quantitative analysis for chemical sample is best performed with the use of a internal standard. Ayan. So, what standard is being used? In here, we make use of a control that is diluted with an internal standard that is known as a yttrium. Ayan. So, yttrium po ang madalas na ginagamit po natin as a calibrator sa quality control ng ating pong aparatus ng ating instrument. So, madalas na ginagamit po natin na standard is the yttrium. Ayan. Then we have the quadruple mass spectrometry. This is the picture of the quadruple mass spectrometry or quadruple mass spectrometer. This typical mass spectrometer is the one that is commonly used in the ICP-MS. So, kung pinag-uusapan po natin kanina si ICP or inductively coupled plasma, madalas daw na ginagamit na MS is the quadruple mass spectrometer. The analyzer consists of 4 parallel conducting rods. Ayan, 4 po sila ng mga rods that are arranged in a square array. Applying for the RF o yung radio frequency and constant DC o yung direct current voltage to the rods, the instrument can be turned so that only ions of specific will be determined. So dito na, kung titingnan natin, pwede daw natin pong galawin. Okay, yung radio frequency and the current voltage para kung ano lang yung gusto nating i-determine na trace element, yun lang ang kukunin niya. So, kung titingnan natin yung ating picture, it has a source, it has a non-resonant ions, ayan, kung nakikita po ninyo, and we also have the resonant ion, followed by the detector, and then yun nga po, yung importante dito na napapalitan natin, na-adjust natin yung DC and AC voltages o yung ating pong radio frequency and the direct current voltages, kung saan na-adjust natin based on the desired trace element that we want to get. So, it may pass through the detector. And again, ito po yung ating pong ratio na kinukuha. Yung ating pong M, which is the mass, and Z, this is your ion. Diba? So, ito po madalas ginagamit po natin, lalong-lalo na pagdating dito sa mass spectrometer. This type of instrument tends to be relatively simple to use and maintain kasi nga, di ba, nagagalaw po natin yung radio frequency and the current or the direct current voltage doon sa gusto po nating ma-determine na specific ion. Ngayon, meron din po siyang mga limitations. The resolution or the ability to discriminate between closely spaced mass o yung charge and ion is limited. So, pagdating po sa resolution niya, medyo limited yung mass spectrometer. Being able to resolve the peaks separated by one charge per ion, but not able to resolve the peaks that were separated by a small fraction of a charge over ion unit. Ayan. So, yun po yung limitation niya pagdating sa resolution. So, nahihirapan siyang mag-discriminate closely kapag sobrang dikit na po yung mga ratio nila. Kapag sobrang dikit na yung charge and the ion values, nahihirapan na po itong ating pong quadruple mass spectrometer. Pero yung maganda nga dito, nagagalaw natin siya, na-turn natin kung saan yung gusto nating i-measure. But when it comes to resolution o yung pagdikit-dikit po ng mga values, nahihirapan po itong ating pong instrument. We also have the high resolution mass spectrometry. This is the picture. So, napakalaki pong instrument, napakalaking aparatus. So, other ICP-MS, so sabi natin kanina yung quadruple mass spectroscopy, yun ang ginagamit. Meron ding iba, which is a high resolution mass spectrometer. So, these are usually double-facing sector field instruments. Such instrument separates ions of different, eto nga po, charge to their ion values via the deflection in magnetic field, with ions greater being deflected to a lesser degree to those of the lower MZ ratio o yung tinatawag nga po natin na ion versus the charge ratio. The magnetic field is adjusted to allow only ions of selected to reach the detection system at any given point in time. So, yung ating pong charge to ratio po natin o yung charge to ion ratio. A second device known as electrostatic analyzer corrects certain non-ideal effects, allowing the instrument to achieve high resolution. Commercially available resolution ICP-MS are capable of resolution that is up to 10,000 resolution. So, ang maganda dito, double focusing siya. Unlike doon sa quadruple mass spectrometer natin, ito, pwede mo rin siyang magalaw. Meron din siyang pang-turn doon sa gusto nating makita. At the same time, meron siyang isa pa na tinatawag nila yung second, di ba yung kanina, second device which is known as electrostatic analyzer. Kumbaga, kinokorek niya. So, yung mga certain interferences such as the non-ideal effects, kinokorek niya in order to achieve a higher resolution. So, yun ang kagandahan sa kanya. Meron siyang pangalawang device which is the electrostatic layer that corrects non-ideal effects para mas mataas yung makukuha niyang resolution. The magnetic sector instruments are not able to resolve the elemental isobaric interferences which would require resolution that is much higher than 10,000. So, ang kaya lang po niyang i-measure na resolution is up to 10,000. So, this is 10% valley. Pero yung higher daw ng 10,000, hindi na po niya kayang i-measure. Ayan.